Would you believe me if I told you that within the next 5 years, pills and inhalers won't contain pharmaceuticals or drugs, but will rather contain genetic material that will be able to activate your own body's repair system to fix the mutations? What was once a fantasy is now rapidly becoming a reality. What was once unbelievable is now truly believable. Welcome. I'm Kmiec, the director of CRISPR's Gene Editing Institute. Today, we're going to talk a little bit about the revolution that's going on in human genetics. The ability for us to use genetic tools to correct mutations that are responsible for genetic diseases. The structure of DNA is flexible, and it allowed geneticists and molecular biologists and scientists in general to manipulate its form and its structure. And that's very important because if we're to correct or fix genes, we have to be able to twist and manipulate the DNA in such a way that that is made possible. Our dream has always been, for many, many years, to be able to take DNA and change it in the way that we want to. And that can be for all kinds of things. Making better crops, more efficient pharmaceuticals, and ultimately for curing genetic disease. In my mind, the basis of human gene editing began with obscure experiments done by a geneticist known as Fred Sherman, working at the University of Rochester. And what Sherman did was to take synthetic pieces of DNA, which he himself made in his laboratory, and introduce them into, of all organisms, baker's yeast. Baker's yeast was used because it was a simple organism, but surprisingly, the genes that are involved in the repair of DNA, when DNA gets broken or changed by sunlight, by X-radiation, or by some sort of chemical insult like alcohol or smoking, the same genes that operate in yeast are very similar to those that function in human cells. Sherman's pioneering experiments taught us that it was possible to add synthetic pieces of DNA into yeast cells and change yeast genes. That simple observation set the basis for the discovery of what we now call DNA Band-Aids that help us repair mutations that cause diseases such as sickle cell disease, cystic fibrosis, Crigler-Najjar disease, and even certain forms of cancer. We've known a lot about the repair systems that are operating in our bodies. Every time you go to the beach and sit in the sun, every time you're outside, the cell's normal repair system are functioning to keep you healthy because those insults, that radiation, disturbs and sometimes breaks your DNA. And that breakage needs to be repaired, or the cell would die or be transformed into a cancer cell. So we've always known that the cell itself, the human cell, has the capacity to repair itself, but it needs to be told what to repair and where to repair it. One of the most important parts of any scientific endeavor is to understand the basic mechanism of action. This is somewhat obscure work and generally doesn't make the headlines, but it's the foundation of the basic biochemical and molecular mechanisms that lead us to major discoveries that can eventually be translated into the clinic. Sherman's work prompted laboratories like my own to study how do these little pieces of DNA actually function? How do they actually work through and make their very specific changes in DNA? And for a period of about 10 years, from the late 90s to the mid-2000s, it became very evident that this effect would not only work in yeast organisms or baker's yeast, but could also be translated into human cells. So those experiments carried us forward for quite some time, and people from all over the world began to look at how it is that such little pieces of synthetic DNA, synthetic band-aids, could actually evoke this remarkable change in the DNA. So why isn't sickle cell disease and all the genetic diseases not yet cured? Well, it didn't work efficiently enough. It worked in about 1% of all of the cells that we tried to treat, but ultimately, to have a clinical effect, it must work at a much higher frequency. We started to look for ways in which we can improve the efficiency. How could we elevate the gene editing, the gene repair reaction using these synthetic band-aids? Now, we had known for a long time by colleagues of ours that were working in the space of DNA damage response. This is an area of science where people try to understand why you simply don't get cancer or why you don't die when you walk outside and get bombarded with UV light or get a sunburn. The body really wants to help you despite our own intentions. Fundamentally, the body is trying to protect you against yourself. This area told us something very important, and that if we broke the DNA, the natural repair systems in our body actually elevated, and the ability to fix DNA went up. So how could we ramp things up using these synthetic band-aids? We decided to test out a mechanism by which we would intentionally break the DNA at very low doses of radiation, or by using anti-cancer drugs that also serve to break the DNA. And lo and behold, it worked. The frequency of gene editing directed by these synthetic band-aids went up almost 10, 20, 30-fold. But how do you specifically tell the agent where to break the DNA? Because the fundamental problem with radiation and anti-cancer drugs is it breaks the chromosomes everywhere. And so while we were seeing an improvement in gene editing directed by the little patch of DNA, the other pieces of the chromosome were also getting broken. And that serves no good. Although we were able to increase the frequency of gene repair or gene editing, the damage that was caused in the human cell, the breakage of the chromosomes, the non-specific destruction, could not be viewed as being clinically relevant and certainly could not be translated into patients. So we needed something else. Strangely enough, a group of scientists were working on the fermentation of milk. How does milk spoil? This happened in the 1980s in Europe. And they were studying a very unusual bacterium known as lactobacilli. Scientists studying the bacterial species lactobacilli made a fascinating discovery when they looked at the DNA in the chromosome. They found that there were segments of the bacterial chromosome that were not bacterial DNA, but rather viral DNA. And it was suspected that these viral DNA segments came into the bacteria as previous infections. Curiously, these viral DNA elements were repeated and strewn throughout the chromosome, and they came from various sections of viral DNA that had been known previously. It was known for a long time that bacterial cells could in fact be infected by viruses. Viruses we know as bacteriophages, and they come in all shapes and sizes. Scientists discovered that these viral sequences inside the bacterial cell actually have a function. They contribute to a primitive immune system that protects the bacterial cell against subsequent viral infections. So how does a bacterial immune system work? It turns out that these bacterial cells use the viral DNA segments incorporated into the chromosome to build antiviral complexes. Complexes we now define and know as CRISPR. CRISPR refers to DNA sequences embedded in the bacterial chromosome that are clustered, regularly interspaced, short palindromic repeats. The viral sequences provide the bacterial cell
the capacity to respond quickly to a secondary infection, very similar to the way we respond to secondary infections with a built-up store of antibodies, perhaps against flu or COVID-19. So with all our understanding about how CRISPR actually works, where it came from, and how it can edit human DNA, why don't we have treatments now for all inherited diseases using a gene editing toolbox? The answer is that nature is more complex than we once imagined. As wonderful as the CRISPR genetic tool is, it is a little bit imprecise. That means that while we design it carefully to target a specific site or mutation that causes the disease, it can also act at a second site, or it can be a little bit unfaithful and leave a genetic footprint behind where it actually is intended to work. We call that collateral genetic damage. One of the most important areas of CRISPR research now is to investigate how that happens. How can scientists make the CRISPR gene editing tool more precise and less imprecise? One of the scientists that's involved in unraveling the mystery of how CRISPR works in terms of precision and imprecision activity is Dr. Brett Sansbury, who works at the ChristianaCare's Gene Editing Institute. Brett and her team have investigated what we call the diversity of genetic outcomes. In other words, when CRISPR enters a cell, it leads to a series of different products that may or may not have deleterious effects. So I've asked Brett to join me today and talk a little bit about the system she developed to study this remarkable phenomena, the diversity of genetic outcomes. Brett?
Much of our focus in the discovery group is based around CRISPR mechanisms and how CRISPR gene editing both imprecisely and precisely repairs DNA. Many of our projects in the discovery group actually focus around using an in vitro gene editing system to further characterize and define many of these gene editing tools. In this in vitro system, we have been able to define things like precise gene editing, strand bias, and interestingly, a diversity of outcomes. Although we see all of these challenges in CRISPR gene editing, precise and imprecise repair, we're still incredibly optimistic, especially with all of the advances that our lab and many other labs are making to better understand these tools and apply them.
So those of you who are interested in science in general, we have a problem for you. Help us redesign CRISPR. Help us move forward to make this genetic tool work more efficiently. As Brett and I work through the fundamental mechanistic study using the cell soup, what we noticed was this model system was so simple, and that's really the key, is that when model systems are simple to use, you get a lot of really good data. And so this model system could also be done by people who weren't necessarily completely familiar with how CRISPR operated. And we developed a genetic exercise, a laboratory exercise for high school students, for college students, to introduce them into the use of gene editing, and we call that CRISPR in a box. This is a laboratory exercise that people can actually use. CRISPR, the Cas protein, the genetic band-aid, the DNA, synthetic DNA molecule, and the cell soup that allows them to understand the power and the interest of using CRISPR-Cas going forward. Remember back when I started with Fred Sherman and his model system in baker's yeast? So after almost 40 years in this field, we are still relying on understanding the fundamental steps of how things work. And clinical application of CRISPR-Cas is going to depend on even more understanding of that. So I'm here to end by challenging you students to join us in the fight against sickle cell disease. Help us understand how to make CRISPR work more precisely. and gain your perspective on how we advance this technology. As we go forward, as we use CRISPR in the clinic, in the next 5 to 10 years, it is our hope that no longer will you be taking pharmaceutical, drugs, and even inhalers to treat genetic diseases, that our genetic toolbox, which we've outlined today, is the one that physicians will give you to treat and eventually to cure these horrible diseases.